Two qPCR reactions can amplify the exact same target, but generate fluorescence in completely different ways. One method uses a simple DNA-binding dye. The other relies on highly specific fluorescent probes. So how do these systems work, and why would a scientist choose one chemistry over another? In this video, we'll explore the fluorescence strategies behind qPCR detection, including SYBR, probe-based assays, and hybridization probes. If you enjoy learning about qPCR, molecular biology, and fluorescence-based detection methods, make sure to subscribe so you don't miss future videos. And if you'd like more structured, step-by-step training, you can also explore our online courses and educational resources using the links in the description below. Let's start with DNA-binding dyes. DNA-binding dyes emit fluorescence only when bound to double-stranded DNA. The dye is added directly to the PCR reaction mix. As amplification progresses, more double-stranded DNA is produced, and the dye binds to these newly formed DNA molecules. Once bound, the dye fluoresces. As PCR product accumulates, the fluorescence signal increases proportionally. The qPCR instrument measures this signal during each cycle, allowing amplification to be monitored in real time. The most commonly used dye in this category is SYBR. Unlike DNA-binding dyes, probe-based assays detect a specific target sequence. This provides much higher specificity and also allows multiplex detection, meaning several targets can be analyzed in the same reaction. Let's begin with hydrolysis probe assays, commonly known as TaqMan assays. A hydrolysis probe assay uses two primers and a fluorescently labeled probe. The probe binds specifically to the target sequence between the primers. It contains a fluorescent reporter dye at the 5' end and a quencher at the 3' end. When the reporter and quencher are close together, fluorescence is suppressed through a process called FRET, fluorescence resonance energy transfer. During the extension step, Taq polymerase synthesizes new DNA and reaches the bound probe. Using its 5' to 3' exonuclease activity, the polymerase cleaves the probe. This separates the reporter from the quencher. As a result, fluorescence is generated and detected during each amplification cycle. Because fluorescence occurs only when the probe binds to the correct target sequence, hydrolysis probe assays provide very high specificity. Several other probe formats are also used in qPCR. These probe designs generate fluorescence using different structural mechanisms. Molecular beacons form a hairpin structure. In the closed state, the reporter and quencher remain close together, so fluorescence is quenched. When the probe binds to its target sequence, the hairpin opens. This separates the reporter and quencher, allowing fluorescence to be emitted. Importantly, the probe is not cleaved. Scorpion probes combine a primer and probe in a single molecule. The probe region forms a hairpin structure that keeps fluorescence quenched. After amplification, the probe binds internally to the newly synthesized target sequence. This opens the hairpin structure and generates fluorescence. Hybridization probe pairs use two separate probes that bind next to each other on the target sequence. One probe carries a donor fluorophore, and the other carries an acceptor fluorophore. Fluorescence occurs only when both probes hybridize close together on the target. This proximity allows energy transfer between the probes, producing a detectable fluorescent signal. Now let's compare DNA-binding dyes and probe-based detection systems. DNA-binding dyes, such as SYBR, are simple, cost-effective, and easy to design. No probe is required, making assay development fast and straightforward. However, these dyes detect any double-stranded DNA, including non-specific products and primer dimers. Because of this, Mel curve analysis is usually required to confirm specificity. Multiplexing capability is also limited. DNA-binding dyes are commonly used for gene expression analysis, screening assays, and experiments where simplicity and cost are important. Probe-based assays provide much higher specificity. Fluorescence is generated only when probes bind to the intended target sequence. This reduces detection of nonspecific amplification and enables excellent multiplexing capability. Probe-based assays are also highly suitable for mutation analysis and SNP detection. However, they are generally more expensive and require more complex assay design and optimization. In general, DNA-binding dyes are simpler, faster to develop, and more cost-effective. Probe-based assays are more specific, more versatile, and better suited for complex applications. The best choice depends on the goals of the experiment, the required specificity, and the complexity of the assay. Different qPCR chemistries use different strategies to generate fluorescence, but they all share the same goal: detecting DNA amplification in real time with accuracy and sensitivity. Thanks for watching, and see you in the next lesson.